Hello, my name's Andrew Traherne and I work for UK Biobank. And I'm here with Professor Rory Collins, who's UK Biobank Principal Investigator. He's the man actually in charge of the project. To find out more about UK Biobank genotyping 500,000 of its participants, news which broke today. Professor Collins, hello.
Hi.
Hi. Why are scientists so interested in genes?
Well, we're interested in genes because they can help us to understand why it is one person gets a particular disease and another one doesn't. They can really indicate causal relationships between various biological factors and the risk of disease.
And how will they be used with all this other data and information that we've got from Biobank participants?
Well, what we're going to be doing is get some several 100,000 markers measured all across the genetic material of each participant. And we can then look to see whether particular variants of each of the markers differs between people who get a particular disease and people who don't get that disease. If we find that the variant of the marker at any particular point in any of these several 100,000 locations is more common in people with disease or not, it tells us that that bit of the gene, or the genetic material I should say, is important for that disease. And then we can narrow down to find out what it is that's going on there, what mutation, what variation has occurred and why that is causing disease.
And just to be clear, it's not then just what we know of as the genes which seem to tell cells how to operate. It's lots of other information between the genes that might be of importance.
That's right. So what we're measuring are markers, and they may be close to bits of the genetic material that are related to the risk of disease. They can be inside the genes, and the genes are what makes the proteins that then help in the function of the body, or they can be bits in between the genes that actually influence the way in which the genes produce the proteins. So it could be anywhere throughout the whole genetic material.
Gosh, it sounds like a very complex and complicated process.
There is this bit of luck that because we inherit not just each bit of the genetic material from our parents, but we inherit lumps, we can use these markers to tell us about variation around the markers. That's why instead of having to measure 3 billion markers, we can get away with measuring some hundreds of thousands of markers. There's even a nice thing that we can use information that's emerging from sequencing, where all 3 billion markers are measured, to actually estimate what the variations are around the markers we measure. So we can get even more information by combining these data and therefore get even more precise associations between bits of the genetic material and various diseases.
Now, should participants be at all concerned that this is somehow providing very personal information about themselves, which they may want kept secret or something like that?
There are a lot easier ways of identifying people. If we were to give names and addresses out, that would be much more straightforward. And the genetic material is being provided for the assays completely anonymized. The data will then come back and researchers will be able to use the genetic data and all of the other data that of course has been obtained from participants, from their questionnaires, from measurements, from linkage to health information, but it will be used by bona fide researchers and it will be anonymized when they use it.
And with regard to protecting people's anonymity and ensuring that scientists don't try and find out who these participants are, I mean, is it just a matter of trust in the researchers, or are there other systems in place to make sure that scientists really can't do that?
Well, it's a terrible thing to say, but the reality is researchers aren't interested in individuals. So, I mean, there isn't a drive to do that anyway. But there are lots of protections that we have in place. First of all, the data that the researchers get has identifying information stripped off it. So they only get the data they need to do their bit of research. And we have controls in place where the researchers have to be approved and they have to sign a contract to say that they will not try to identify participants and that they will keep the data secure themselves. So we have a lot of safeguards in place.
And just looking to the future now, I mean, what sorts of research do you think might benefit particularly from this sort of development?
Well, to give one example, there is a new class of drugs that's being used to, or being tested to lower cholesterol. The genetic underpinning of that drug was only identified within the last decade. It's gone from the identification to actually being tested in late-stage trials, likely to be, if it's positive, be used quite soon. So the hope is that this kind of work will identify those kind of causal links between genetic variation and disease, not just for heart disease, but for many other conditions that have not been well studied: dementia, joint diseases, hearing loss, eye disease, all of the kinds of things that were examined at the baseline visit and that we can follow up through the health record linkage that participants have agreed to.
Well, and as a participant, I was very excited to hear about this particular development. And I would say to participants watching this on the website that I hope that answers a few questions. If you have any more questions or interest in this particular piece of research, please do make contact with us either by website or on the phone. The details are on the website. And we'll do our best to answer those questions for you. Thank you.